Ooh, I know. Let's talk about evolution from the perspective of predicting changes in allele frequency. It's me, Moser, and after this video, you should be able to use information about inheritance patterns and conditions in the environment to predict changes that will occur in allele frequency. So you should already know what alleles are, and you should have a basic understanding of what allele frequency means. Awesome, let's start. So you'll remember that the last two definitions we've worked with of evolution are that we can talk about evolution as a change in the distribution of a trait in a population, or, more importantly for our purposes here, we can talk about evolution as changes in the frequency of an allele in a population. That's what we're going to focus on now. We're going to go back and review some basic genetics to put that whole change in allele frequency thing in context, okay? So you should remember from middle school that traits that are controlled by a single gene, we call these monogenic, that have a simple dominant recessive inheritance pattern. We show these as having the dominant allele with a capital letter and the recessive allele with the same lowercase letter. Now, all organisms that are the result of sexual reproduction have got two copies of each allele. They got one from each parent. And those two alleles, like this, make up their genotype. So we know their genotype are the kinds of genes they have, but we can determine the physical expression, or phenotype, of the gene combination, or genotype, that any organism has. For simple, dominant recessive inheritance patterns, having even one copy of the dominant gene means that the organisms show the dominant version of the trait, and the recessive version is hidden. The only way to show the recessive version of the trait is to have two copies of the recessive allele. Is this ringing a bell? Oh, guinea pigs. So to show an example, in guinea pigs, smooth fur, big F, is dominant to a wavy furred coat. A guinea pig with a big F genotype would have smooth fur, as would a guinea pig with a big F little f genotype. The only way for a guinea pig to show a wavy coat, or to have the wavy phenotype, would be to have a little f genotype. So is there any survival advantage to having a smooth or a wavy coat? Probably not, but let's imagine. A vicious predator hunts wild guinea pigs in the mountains of South America. And there actually are wild guinea pigs in the mountains of South America. Go ahead and look it up. Smooth-coated guinea pigs slip right out of its clutches, too slick. But those wavy-coated guinea pigs. That wavy coat provides just enough texture to let the predator grab and hold the helpless piggies. Which genotype will be captured and killed more often? Since only the wavy guinea pigs are being killed, and to have a wavy coat, a piggy must have a little f genotype. Little f genotype guinea pigs are the ones dying in the clutches of our vicious predator. Crunch, crunch. What does this mean for allele frequency? If we assume that our population started off with an even mix of genotypes, which actually gives us more straight-furred guinea pigs, the dominant phenotype, which we often expect to see more of, we would see an allele frequency of 50% for the recessive allele. And here it is on our chart. If only or mostly little f guinea pigs are being killed, we would expect to see a decline in the frequency of the little f allele. Will the little f allele disappear from this population? No. Why not? Because there are plenty of dominant phenotype, or straight-furred guinea pigs, carrying a copy of the recessive allele. These individuals have one copy of the dominant and one copy of the recessive allele. We call them heterozygous. More about that later. This means that the little f allele stays in the population, even if its frequency declines. You can think of it as a hidden F. Would the change in allele frequency be different if the dominant phenotype was being killed more often? Let's think that one through. Imagine we now have a predator who, instead of eating the little wavy-furred guinea pigs, prefers to prey on straight-furred ones. Remember that straight-furred guinea pigs can have a genotype of either big F or big F little f. Since straight fur is dominant, it only takes one big F allele to show the straight-furred phenotype. Assuming that we start with the same even mix of straight and wavy fur, we should see the same allele ratio of 50% big F, 50% little f. In this case, let's look at the frequency of the dominant allele. If our predator is eating straight-furred guinea pigs, that means they're eating animals with big F, and big F, little f genotypes. The big F allele is clearly going to become less common. Since there are no hidden big F alleles, having a single big F allele, single dominant allele, means you show the dominant phenotype. We can expect to see a big decline in the frequency of the big F allele. Is the big F allele at risk of disappearing from this population altogether? Yes, it is. Crunch. Now, let's talk about how this might look different if the trait in question is not controlled in a simple dominant recessive pattern. Some traits are controlled by a pattern known as incomplete dominance. In incomplete dominance, there are 3 phenotypes. A flower with incomplete dominance with the alleles R for red, big R shows a red flower, and little r for white, little r shows white flowers, would show a phenotype of pink for a genotype of big R, little r. Whoa, how cool! We use as our example a species of moth whose wing color is controlled by a gene with incomplete dominance, where big B produces a moth with a dark brown wing, and little b produces a pale tan, nearly white moth. But big B, little b produces a plain old middle-of-the-road medium brown moth. If this species is living in a location where most of the backgrounds they might land on are very dark, it's likely that predators, like this guy, will have an easier time picking out the lighter moths. Crunch, crunch. What will this do to allele frequency? So let's assume that we started off with an even mixture of phenotypes. About a third of the moths have dark brown, a third have medium brown, and a third have pale tan, almost white wings. How do we figure out the allele frequency for these individuals? I'm telling you that it's about 50% for each allele, but let's prove it. For any allele, the frequency of that allele is just the number of alleles of that particular kind divided by the total number of alleles in the population. So for big B, the allele frequency would be the number of big B's divided by total alleles, and for little b, it would be the number of little b's divided by the number of total alleles. We're going to start off by assuming that we've got a nice, easy to work with number like 10 of each of these phenotypes. So if we have 10 dark brown moths, phenotype big B, and we take our big B, and multiply by 10, that means we have 20 individual large B alleles. Okay? Well, if we have 10 of the medium brown moths, 10 big B, little b individuals gives us 10 copies of big B and 10 copies of little b. And if we have 10 of the nearly white moths, that's 10 little b individuals for a total of 20 little b alleles. If we add that all up, we get a total of 60 alleles. And we can double check that by saying, well, we had 30 individuals with 2 alleles each. Yeah, that should be 60. So we have 60 total alleles. How many big B's do we have? Well, 20 and 10 gives us 30. 30 out of 60 means we have a 50% allele frequency for big B. Same goes for our little b. 30 little b's, 10 plus 20, out of 60 give us a 50% allele frequency. The moths being eaten most often are genotype big B, and little b. The survivors are big B. What change can we expect in the frequency of the little b allele? Well, it's going to decrease in terms of the percentage of population that carries the little b. Individuals with little b are being eaten. individuals carrying even a single little b are heterozygous individuals. Big B, little b are being eaten. This recessive allele will see big declines. Could it disappear from the population altogether? Yeah, it could. We may end up with a situation where 100% of our moths are big B, big B. What change can we expect in the frequency of the big B allele? Well, it's going to increase in terms of the percentage of alleles in the population that are big B. Even though heterozygous, big B, little b individuals are being eaten? Yes. Think about what's happening to the little b as its share of the total allele frequency decreases. An increasing percentage of the total alleles in this population are going to be big B. Okay, so now we have a sample problem, and I want you to write down the information you're being given. We have some species of plant that exists in a tall and a short form. The height characteristic is a simple dominant recessive pattern where T, big T, tall, big T, is dominant to short, little t. A predator is introduced to the ecosystem which prefers to eat the tall plants. What changes can we expect to see in the allele frequency for big T versus little t? So when we start to work with any problem involving changing allele frequencies, we can follow a set of steps to solve just about anything. Step number 1, check the inheritance type. Is it a simple dominant recessive or incomplete dominance? In this case, we were told it was a dominant recessive inheritance type. Step 2, list all the possible genotypes and the phenotypes that they would produce. So in this case, we have big T, which gives us a tall plant, big T, which gives us a tall plant, and little t, which gives us a short plant. Step 3, determine which phenotypes are being affected. So in this case, we were told that tall plants are being eaten more often. Step 4, translate that into understanding what genotypes will be affected and how. In this case, big T, and big T, little t are being eaten, so they're going to decrease. Based on the genotypes being affected, which individual allele will increase, and therefore which other one will decrease? Will they remain the same in the terms of what percentage of the population carries one or the other? Step 6, double check yourself. Remember that overall 100% of the population has to carry one or the other allele, so when one allele decreases, the other one absolutely has to increase. Okay, do you feel like you can use information about inheritance patterns and environmental conditions to predict changes in allele frequency? Good. You've got practice work to do.